mute. Okay. Okay, so you guys can hear me?
Yes.
Perfect. Any questions about any of the material we covered so far? Okay, so if you don't have any questions, we're just going to go ahead and jump into photosynthesis. Photosynthesis is chapter 10, and you guys probably learned this in 7th grade science, that plants take in light from the sun, and they take in carbon dioxide from the air, and they magically produce food for us. And so we know that that reaction is actually that plants take in carbon dioxide and water. CO2 and water, and we have energy from the sun. And what kind of energy is sunlight? Kinetic or potential?
Kinetic.
Sweet. Yeah, because it comes from the sun, which is really far away, so it has to travel. And any energy in motion is kinetic energy. And so we know we have a balanced equation. We learned this in chapter 2. We had to balance out the equation to make sure that everything is the same on both sides, because we can't create or destroy matter. We can only change forms. And so plants produce glucose and give off oxygen. And that's no secret. The glucose molecules that are produced then are linked together in long chains. And what do we call long chains of glucose? And we learned this in chapter 5. So long chains of glucose that are made in photosynthesis are called, you guys would call them carbs, but the real name is amylose, because all sugars end in OSE. So plants are making glucose, they're linking it together, they're storing it as carbs. And then remember, we talked about glyphosate, so that plants can take their amylose or their glucose and they can make other things with it. So Roundup, they use this to synthesize proteins and DNA and lipids and all that stuff that we learned about in chapter 5. Plants use glucose as the raw material to do all of this. So if you spray Roundup on your weeds or your plants, it interferes with their ability to turn glucose into proteins and they die because plants don't have a way to get proteins. They can't get it from the air. There's no protein in dirt. They have to build these things with enzymes that we learned in chapter 8. So that's what's going on, and this is the balanced equation. And what you guys learned in chapter 9 is the exact opposite. So that's respiration. And we take in oxygen and glucose, and we learn we can use other things besides glucose, like proteins and things like that. And we, instead of needing energy, we give off energy. We produce energy in the form of ATP, and we produce water as a byproduct, and you guys saw that in the Krebs cycle. And we also produce CO2, and you also saw that in the Krebs cycle. And when we converted pyruvate to acetyl-CoA. So if you know respiration, you know photosynthesis, because it's the exact reverse of that reaction. But the question is, what does this arrow mean? And that's what this whole chapter is about. Just like respiration, what goes on with this arrow is a lot more complicated than this relatively easy equation. All right. So before we start into this, we need to talk a little bit about energy and how nutrients are made. And so these are just some terms that you guys are going to need to know. So auto means self, trophic means nutrients or food producing. So an autotroph organism is something that produces its own food, right? And that could be using sunlight, which is photo as light, like photography. So there's photoautotrophs, and they use light as an energy source. I'm sure you know many examples of this. We've talked about plants. We've talked about cyanobacteria in chapter 1, and algae as well when we were doing classification. But and up until we were able to discover deep sea organisms, we didn't really know this existed. But when technology advanced enough to explore the deep ocean, which was after we put a man on the moon, we discovered that there are these things called black smokers or hydrothermal vents at the bottom of these deep trenches in the ocean, several miles deep, and that they contain an archaeobacteria. Remember, we talked about this as archaea is old or ancient bacteria, and they use chemicals, specifically hydrogen sulfide, to produce sugars. And we're not going to get into the specifics of this. It's just a different pathway, but this is quite the find back in the day, because this tells us that things like the icy moon of Jupiter, Europa, even though it's covered in miles and miles of ice, underneath it there's liquid water, and that water, and it has a solid core, so that means that it can have these smokers at the bottom of its ocean, and without any sunlight it can still have life. And so that's why we're really interested in this moon of Jupiter, because we know that water is densest at 4 degrees Celsius. We know things can live at 4 degrees Celsius, like crabs at the bottom of the ocean. And we know that it's possible that we could that living things can use chemicals instead of light as an energy source. So that's why we're really interested in looking for life on these icy moons of these supergiants like Jupiter and Saturn. And they're going to spend a few $1 billion to explore this relatively soon, in the next 10 years. Okay, so these are the modes of autotrophs. These are the ways that you can make your own food. You could use light, you can use chemicals, The only thing we know that are chemoautotrophs are these archaeobacteria, but we don't know what we're going to find on other worlds. And then if you can't make your own food, then you're like us, like humans, and that means that you have to use you rely on other organisms for your food. So animals, fungi, things like that. And we learned about these kingdoms in chapter 1. Those are heterotrophs. And so we have auto or heterotroph, and then within auto we have photo or chemi autotrophs. All right. Any questions so far? All right, so with that said, what we're going to focus on is the photoautotrophs for this chapter. And as far as we know, on Earth, all photoautotrophs use these organelles. It's an organelle. We learned about this in chapter 6, called the chloroplasts. Remember, plast are terms we use for organelles in plants, and chloro is the term for the pigment that's found within it. When that pigment is called chlorophyll, so it's green. You guys know plants are green, and chlorophyll's job is to absorb light energy to create photosynthesis. So when the sun shines on chlorophyll molecules, these pigments molecules, the light energy actually excites the chlorophyll molecule. I'll show you how this works. There's actually a magnesium in it. And do you guys remember from chapter 2, whenever we put energy on an atom, what happens to the electrons? So in chapter 2, we talked about when we put light energy on an atom, it excites the electrons and they go to a higher energy level. So they move away from the nucleus. And the more energy we put in, the further away they go. So imagine if you had a rocket and you launched it up into, let's say, a couple miles into the stratosphere of the Earth. What would you expect that rocket to do when it ran out of fuel, if it didn't get to outer space?
Would it fall back down?
Right. And so that's what normally happens. When we add energy, the electrons jump, kind of like a rocket ship. And once that energy dissipates, then it falls back down and that energy is released. Usually as light energy or heat energy. And if you guys don't remember this, you can go back to chapter 2 and check it out. But if we kept putting energy in that rocket and we kept putting energy in that rocket and it got further and further and further away from the Earth, what would you think? What would happen eventually to that rocket?
Would it float away?
Yeah, it would just float away. And so that's what's going on here. The sunlight is going to shine on this magnesium, and it has 2 electrons in its outer shell. And those electrons are going to go higher and higher, and eventually they're going to float away. And what do we call flowing electrons? We learned about this in chapters, the very last chapter, in chapter 9. What's that called? You use it every day to charge your cell phone.
I'm sorry, what was the question?
What are flowing electrons called? You use it every day to charge your cell phone. So flowing electrons is called electricity, and you guys know that we can do a lot of things with electricity. That's part of the electron transport chain. That's how we make ATP. And we're going to use these flowing electrons from the exciting chlorophyll to generate electricity to do work as well. We're going to make ATP. We're also going to have electron carriers, because remember, you don't want electricity flowing just freely inside a living system. That's not safe. So anyway, we'll cover this in a second, but I just want you guys to realize, to remember that when we put energy on an atom, it causes the electrons to jump to a higher energy level. And the more energy we put in, the further away it goes. Eventually it's going to float away. And once it does, once it starts flowing electrons, it's generating electricity, and we can use that to do work. And if you don't believe me, go outside and turn off your fuse box and see how many things you use electricity for. All right, so chloroplasts, these are the organelles within the, you don't need to know this, but it's called mesophyll. It's a layer of leaves. If you tore a leaf in half and looked at it under a microscope, you would see that these cells, this area in here, are mesophills, and inside them are chloroplasts. You do need to know that plants have mouths. We call those stomata. That's where they do gas exchange, just like you guys breathe in oxygen and breathe out carbon dioxide. Well, they breathe in carbon dioxide and breathe out oxygen through these stomata. So that's a word that you need to know for this class. And that's where gas exchange occurs. And then the sugars and things that are produced flow through veins and plants. Plants don't have hearts, so this travels, like we talked about in chapter 3, through evaporation or cytoplasmic streaming. So we talked about this in chapter 3. And then we talked about cytoplasmic streaming in chapter 6. So that's how plants make sugars and how they transport it to other cells that aren't sugar producing cells. Just like the stems and the roots and things like that, because the roots don't get sunlight, so they can't do photosynthesis. Any questions about this so far? All right, so this is a cross section of a mesophyll cell. And you can see, just like mitochondria, we can have lots of chloroplasts in each cell. You know, this cell looks like it has maybe 30 or 40 chloroplasts in it. And that means that it's producing a lot of energy and sugars, because that's the goal. So this is a cross section, an artist drawing of a chloroplast. Just like a mitochondria, it has an outer membrane and an inner membrane. That's shown here. Inner membrane, outer membrane, and the space in between it is called the intermembrane space. So just like the mitochondria, exactly like the mitochondria. Outer membrane, inner membrane. Inside here are these little, look like coins, and we call these thylakoids. So these are thylakoids, and they're stacked on top of each other like a roll of quarters, and we call these collectively grana. Sorry, I wrote over that, but that's the inside area of the chloroplast is called the stroma. This is equivalent to the matrix, which is the inside part of the mitochondria. So if you know the mitochondria, you pretty much know the chloroplast. The only thing that's different is they have these thylakoid discs that are stacked on top of each other, and those are called grana, and you just need to learn that. And that the inside part is called the stroma instead of the matrix, which it was called in the mitochondria. And this is just parts of it. The inside of the thylakoids, so the inside part here in the thylakoid itself is called the lumen of the thylakoid. And we covered this again. We covered a lot of this in chapter 6. So this is probably just a review, or hopefully it's a review. Okay, so in photosynthesis, we have 2 stages of photosynthesis. This is the representation of that as guarana, right? And each one of these is a thylakoid disc. And so what happens is the light comes in, right? It strikes the chlorophyll in the thylakoid. It excites the electron. We flow electricity, and it takes water and it splits it. So it's going to split water. And by doing that, it produces oxygen. So we really need 2 waters. So we have 2 H2Os, and that produces 2 O2s. There is it. So we have 2 waters that produces a single oxygen. So we have it balanced, 2 oxygens and then 4 hydrogens. So we're going to produce 2 H2s, because remember, oxygen and hydrogen are always going to be combined together, because this one only has a single electron, and it needs 2, and then oxygen produces those double bonds. So any free oxygen then encounters another oxygen is going to form a double bond. And we learned about this in chapter 2. So you can go back and review that if you're not sure about it. All right, so plants take in water, they split the water, that produces oxygen and hydrogen. And we can split this hydrogen just like we did, remember in glycolysis. Where we took 2 hydrogens off of glucose, and we took 2 electrons from those 2 hydrogens. Remember, because the hydrogen has 1 electron and 1 proton, and we use those electrons for the electron transport chain. So if you don't remember that, you can go back to chapter 9 and check it out. But it's true. The enzyme is called dehydrogenase, and when it does that, it does an oxidation reduction reaction. So remember, glucose loses electrons. So what is it? Oxidized or reduced?
Oxidized.
So
Reduced.
you're right the first time. So when you lose electrons, you're oxidized. Remember, Leo the lion goes grr. So lose electrons, you're oxidized. If you gain electrons, you're reduced. So glucose is losing electrons, right? So it's oxidized. And then what's gaining those electrons? Do you guys remember what the electron carriers are called? So I'll just remind you, but you can go back and check it out. So NADH is an electron carrier, and FADH2 is also an electron carrier. So those are the things that are getting reduced because they're gaining electrons so that they can transport them safely through the cell. So that's what's going on in the light reaction. And we're going to break this down. During the light reaction, we produce ATP. Remember, when we were doing glycolysis, we produced ATP. And then we also produced electron carrier called NADH. This is a little different in plants. It's called NADPH. Why do you think it's called that?
Ribophosphate.
Yeah, you're right. So it has 1 extra atom in it that's a phosphate. Nobody knows why plants evolved this method instead of NADH, but it works exactly the same. So we're making an electron carrier, and we're making what we can use directly as energy. these two things are going to get pushed into so this is the inside of the chloroplast. Remember, we call that the stroma. And that's where the Calvin cycle occurs. Just like, remember we talked about the Krebs cycle? And this guy Krebs named it, and then people got pissed off and they changed it to the citric acid cycle. So same thing here. This guy Calvin claimed he discovered it. Other people disputed it. So now they just call it the dark reaction, usually. And the reason it's called the dark reaction has nothing to do with the fact that it's evil or anything like that. It simply means that it doesn't need light to run. So it can run in the dark. So this is also called the dark reaction. I'm going to use Rx for reaction. And the dark reaction needs energy in the form of these NADPHs. Remember, electrons have energy, and ATP is our direct energy source. Both of these are going to be used in the Calvin cycle. And in the Calvin cycle, what happens is plants take in carbon dioxide out of the air, and they combine it with a sugar called ribulose biphosphate. This slide's getting super busy, but what you need to know is, so ribulose is similar to ribose, like in DNA. So do you remember how many carbons are in that sugar? We learned that in chapter 5. So it's a pentose. And what does biphosphate mean?
2 phosphates.
Yeah. So it's a pentose sugar, like DNA and RNA, like ribose and deoxyribose, that has 2 phosphates on it. And that plants use this to add a carbon. And when we add a carbon to a pentose, what do we make? So instead of 5 carbons, it has 6. So it becomes a hexose. So we're fixing the carbon dioxide in the air into sugars, and then it spits out a single carbon, which we would call a monos. And that single carbon sugar is glyceraldehyde. Well, it's going to be turned into glyceraldehyde 3-phosphate, but I'll show you how that works. Because it's a cycle, it has to regenerate this ribulose biphosphate, and so it has to return 5 of the carbons back to the beginning. It can only expel 1 carbon that it's fixing out of this single carbon from carbon dioxide, and I'll show you that. So takeaway from this is the Calvin cycle is also known as the dark reaction. So it has 2 names, just like Krebs has 2 names. That's fixing carbon dioxide in the air, and then it reduces it, so it's adding electrons. Where do you think the electrons come from that's adding to this carbon dioxide?
NAD. Sorry, I can't see it.
Yeah, NADPH, exactly.
Yeah.
Which we're making in the light reaction, and it produces this single carbon sugar that, you know, we know that you can, if you have a single carbon sugar, it's like a Lego block, and you can add 3 together, and that'll give you a glyceraldehyde 3-phosphate. You add 6 together, and you get a glucose. And so plants are building glucose molecules that they're going to link together into carbs that they're going to store as an energy source. They're not doing it so that you can come and, you know, sneak up on the carrot and take its sugars. They're doing it so they have a way to survive when light isn't present. Just like if you had solar panels on your house, if you didn't store any energy at night, you wouldn't have any energy. And so, but in plants, if they didn't have any energy, it would be like holding their breath. So plants can't hold their breath overnight, they would be dead. So they have to store these sugars so that they can use them, kind of like a power wall, that, you know, a battery that you would use on a solar panel, so you could store energy during the day and then use those batteries to power your house at night until the sun came back up. And then remember, in the light reaction, we're simply splitting water to produce oxygen, and we're making ATP and NADPH. So we're going to get into each of these and the specifics of it right now. Okay, so this is just a sort of a breakdown of what's going on in the light reaction. So light reaction occurs in the thylakoids that have chlorophyll. Remember, this is that green pigment that makes plants green. Remember, the thylakoids are, you know, in the inside of the stroma. Those are those little round discs. And they're inside chloroplasts. That's what this whole thing is a chloroplast. It reduces NADP plus. So this is just like, remember we showed in NAD plus in chapter 9. Same deal to NADPH. Only difference is it has this phosphate in plants. So like I said, similar to respiration. So we're going to add an electron. Actually, we're going to add 2 electrons. And that's why if you gain an electron, you're reduced. So if you add, gaining, adding, same thing, it's going to cause you to be reduced. Oxygen is given off as a byproduct of splitting water, and you guys know plants give off oxygen. And then it generates ATP by adding a phosphate to ADP, just like ATP synthase. Remember how it did that? So we had the phosphate plus ADP, squashed it together, and we ended up with ATP. And you guys know by now, hopefully, that's our energy currency. So that's what's going on in the light reaction. To understand how this works a little bit, you have to understand what light really is. So light is part of the electromagnetic spectrum. If you haven't had physics, this is a quick physics lesson. So there's 4 forces in the universe. One is gravity, which we're all familiar with. Nobody really knows how gravity works, but we do know it exists. And then we have the weak force, which helps hold molecules together. And then we have the strong force, which holds the subatomic particles together. And then we have electromagnetism. You're familiar with gravity. You're really not messing around with the weak force or strong force unless you're doing. So this would generally be chemistry. This would be physics, where they, you know, take super colliders and physics and smash things together to see what parts come out of it. And you're familiar with this. So electromagnetism is simply energy in the form of a wave. And this is a terrible wave, but everyone's seen waves, and you know there's a crest of a wave and there's a trough of a wave. And so the wavelength of this electromagnetic energy is designated by this symbol, which is gamma. And this is what we call the wavelength. You probably know about this, right? Like you throw a pebble in water, it makes small waves. You throw a big boulder, it makes big waves. And you can see those waves, but electromagnetic waves, generally you can't see them. So the wavelength causes it to be different than any other waves. It's still the same energy, it still travels at the speed of light, but it's the length of the wave that makes it different. So radio waves, everybody has at one time probably listened to the radio. Those are electromagnetic waves. They're giant waves. You know, we're talking size of hills or mountains. People have probably used a microwave, right? You use it to cook all the time. Those are a little smaller waves. They're micro. That's why they're called microwaves. And micro, remember, doesn't just mean small. It's a specific number. And if you remember from your metric system, micro is 10 to the negative sixth. Remember, millis 1,000th. So this is 1 millionth. So the wavelength of this is measured in 1 millionth of a meter. And then we have infrared light, which you are probably familiar with. This is like night vision. You can't see really into infrared with your eye, but some animals can, like snakes can detect infrared. Other organisms can see in the infrared spectrum, so they see much better at night than we can use night vision goggles to artificially look into that spectrum, but usually it's invisible to the naked eye. And then we have visible light. So visible light, this is what you see every day. This is all you can see. And visible light falls somewhere between 750 nanometers and 380 nanometers. So which one's bigger, 750 or 380? It's not a trick question. Are you guys there?
750.
Yeah. So on this end, remember, these are the big ones, but we're getting smaller and smaller. But on this end, on the red end, is the 750. And remember, these are nanometers. So we're talking 10 to the negative ninth, or so this is millionths of a meter. This is billionths of a meter. And then on this end would be the shorter ones, the smaller ones, which would be in the 380 range. And so that's what you can see. You can see between 750 nanometers and 380 nanometers. And if we go smaller, we're in ultraviolet, which you cannot see, but you know it's there because it gives you a sunburn. And then X-rays, which you guys can't see X-rays, I'm sure, unless you have superpowers. So, you know, if you could see X-rays, then when you went to the doctor, you would see colors when they, or something, whenever they gave you an X-ray. And then gamma rays are even smaller. These are rays that are given off by like solar flares and things like that. And these are so small. These are in the femto range. So like we're talking 10 to the negative 20th wavelength. And these can pass through the Earth without ever even encountering or interfering with an atom or its nucleus. So they can pass unimpeded all the way through the Earth. So that's basically just some examples of this electromagnetic spectrum. And the reason that this range is actually just a guess, because there are some things that can affect that. So things that can affect one person's ability to see further into the spectrum than another is age. So you guys, you heard of that ringtone that like younger people can hear, but older people can't? Or not, but anyway.
I don't think I've heard of that.
Well, it's true. Do a Google search on sound frequency. There were some shops that had kids loitering around outside, and they started playing this super high frequency sound, and it's kind of like driving bugs away from sound frequencies or dogs. Only young people can hear that frequency, and so as we get older, we can't hear or see as far into the spectrum. So the older you are, the rule of thumb is the less you can see into the spectrum. So that really means that, you know, some people can see violet colors that other people can't, and some people can see red colors that other people can't see at all. Genetics plays a part in this, and sex plays a part in this too. So depending on if you're male or female, females can actually see further into the visible spectrum than males. So females can see things that males can't, believe it or not, and that's true. All right.
That makes sense.
So each range of light, each wave, every color has its own wavelength of light. So I don't know, whenever I was a kid, I would, you know, sit around and think about this stuff, like what makes green green? And what if you told your kid that green was red, and blah, blah, blah. And then I figured out that trick really doesn't work, because these aren't really colors. I mean, we know that they're colors and we see them the same. Some people are colorblind, so they can't see green and red. But these are all numbers. So remember, violet corresponds to a wavelength, which is around, you know, 380 to 350. And red corresponds to a wavelength, which is around 750, you know, and green is around 480, and blue's a little bit less. It's around 410, and indigo would be around 370. And you know, yellow is around 525, something like that, and orange is around 630-ish. So all of these colors actually can be converted into numbers, and to do that, we use a machine called the spectrophotometer. So it's got a fancy word, but if you break it down, it's pretty simple. It's a spectrum, right? Which is what this is, a rainbow, right? And the photo means light, and then we're measuring it in meters, right? In this case, nanometers, but it's still meters, a distance. So a spectrophotometer is something that simply measures electromagnetic energy based on its wavelength. And it can detect microwaves and radio waves and ultraviolet waves, and the spectrophotometer is really the way that you're able to listen to the radio when you get in your car. So that's how light energy works. Do you guys have any questions about that?
No.
Okay. So the pigments in your clothes, they're actually, like if I was wearing a red shirt, so do you think that so let's just for fun, we're going to say red is the red shirt that I'm wearing is at 750 nanometers. So that means that it is either absorbing energy in that wavelength, or it's reflecting energy at that wavelength. So what do you guys think? Is it absorbing or reflecting? Sometimes this is called transmitting.
Red reflects, right?
Yeah, because if it didn't bounce off my shirt to your eyeball, you wouldn't see it, right? If it was absorbing it, you would see black, which is the absence of color. So a black shirt would be absorbing all the colors of the spectrum, where a white shirt would be transmitting or reflecting all the colors of the spectrum. And so your shirt's red because it's reflecting this wavelength. And if it was reflecting 480, then your shirt would be green. And if it was reflecting 750, or I'm sorry, 350, then your shirt would be violet. And you guys know from probably, you know, primary school and colors, you know, there's 3 primary colors. And if you look at your television is actually red, green, and blue. and all these combinations of colors can make all the colors of the spectrum, millions of colors. So you really only need 3 colors mixed together, and you can make any color you want. But that's how we get different colors of shirts, because they're absorbing or reflecting that wavelength. And usually shirts are dyed with pigments, which are the same sort of thing that chlorophyll is. It's a pigment. If I use chlorophyll to dye my shirt, then my shirt would be green, and it would reflect green wavelengths, right? So if it's reflecting green, what is it absorbing?
The green pigment.
Well, it would be absorbing everything but green, right? Like
Oh yeah, yeah.
red, it's reflecting red, but it would be absorbing everything but red. That makes sense? So here's the question. Plants are green, right? Are they absorbing green?
No.
Then that means that they're not using green as an energy source. What are they using as an energy source?
All the other colors.
Exactly. Now, if plants used all of the available energy from light, then what color would they be?
Black.
Yeah. So when we go to these Earth-like planets far away in these distant solar systems, most scientists believe that the leaves of the plants will be black because they would be fully efficient in absorbing all light. Now, that probably makes you go, well, why aren't plants black on Earth? And the answer is nobody knows. Nobody knows why plants don't use green, because they are perfectly capable of using it. There's all kinds of theories and ideas that there were different colored plants on the surface of the ocean that could use photosynthesis, and only in green, and so plants were forced to use everything but green. I've read all kinds of crazy ideas, and some are more logical than others, but in reality, until we have a time machine, we'll never figure this out. I don't think. Maybe, but we don't know. We don't know why plants are green. It's
So
weird.
are the plants that they sell as like black plants, are they just a really dark green then?
I don't think I've ever seen a black plant unless maybe it's dyed.
Okay.
Yeah. So, yeah, most. So we'll talk about the other pigments, like how the leaves change and stuff, and so they shed the chloroplasts because they're going to go dormant, and so they're going to save all their sugars, kind of like a bear hibernating. And so the chloroplasts go away first, but they're left with these other accessory pigments we call carotenoids, which you guys did in the photosynthesis lab. And it includes like beta carotene, and then there's xanthophylls and some other pigments that cause leaves to change color. But I've never seen a black plant that can do photosynthesis.
Okay. Yeah, because I've seen where they sold like a black ZZ plant before, but I just didn't know if it was like a really dark green or if it was actually black.
You know what? I don't know. I'm not really a plant expert, but I'm curious enough to look that up. So I'm going to write that down, and I'll let you know next time
Okay, cool.
what I find out. Okay, so just a reminder that the color that's reflected or transmitted by the pigment is the one that you see. All right, so if you see green, it's reflecting green. If you see red, it's reflecting green. And then it's absorbing all the other colors. Okay, so if it's the experiment that you got, we used to do this in in a live class, but now we're forced to do this completely online, so there's no labs. But the video that you guys watched, basically they take spinach and they grind it up with a mortar and pestle, and I'm sure you've seen those before in like a pharmacy lab, or some people use it to make, you know, cornmeal. So they take spinach, they grind it up, and they can't use water because chlorophylls look a lot like phospholipids, and I'll show you that. And so because they're lipids, they have to be dissolved in something like acetone. So that video that you saw was somebody took spinach, ground it up in acetone, and then they put it on that piece of paper. Oh, this is a terrible drawing. So let me see if I can get green up here, and we'll just simulate this. So, and you guys would have done this in class, though. So the lab techs would have ground up spinach and acetone and give you a bottle of spinach extract, and you just put a dot on here. So you add a drop of it, and you have to let it dry, and you add another drop, and so on and so forth. And you get this concentrated spot, and then you put it in. Use another color. You put it in this petroleum ether, which is a good solvent for fats, and you know it works the same way as if you put if you took a paper towel and put it in a cup of water, right? The water would run up the paper towel. So this is chromatography paper. It's a special paper, but it still acts like paper, and it's going to absorb this liquid, and that liquid's going to travel up. And when it does, when it hits this, your chlorophyll spot, it goes back into solution. So when it's in the solution, it's going to ride this liquid wave up until, and as this is happening, you guys know that these solvents like alcohol and stuff, if you rub ethanol or rubbing alcohol on your skin, you know that it evaporates really fast, a lot faster than water. So what's going on is that all this is evaporating off into the air, and once all of it's evaporated, it's going to leave some of the pigments behind. So so some of the pigments will come out here because they're not as soluble. Change to green. So some of the pigments will come out here because they're not as soluble, and then other pigments will come out here because they're a little bit more soluble, and then other pigments will come out higher up here because they're the most soluble. So chromatography separates things based on their solubility. And we have to do it that way because these different chlorophylls are so similar molecularly that you can't, they almost weigh the same, so you can't use a centrifuge or anything like that. So in a chlorophyll, there's a mixture of different chlorophylls. So there's a chlorophyll A. It directly participates in the light reaction. So this is the one where light hits it and the electrons get excited. Chlorophyll B works like a satellite dish. So you guys know your satellite dish is curved, and you have like a receiver here. And the way that works is it's shaped that way so all the signal focuses on one point. You can do this to like cook hot dogs with aluminum foil or whatever, because you can make something that's really hot. In fact, I will show you. There's a kid who took a DirecTV dish and went to Ace Hardware and got a bunch of magnets. I'm sorry, not magnets, mirrors, and glued it on there. And he called it the death ray. think it's called the death rate 3000 or something. No, 5000. The death rate.
Arizona homeowners, if you're currently paying APS or SRP hundreds of dollars each month.
So I'm just gonna stop sharing this real quick, and I'm just gonna share this video. So the point that I'm making is that if you're focusing all this light energy on one point, it's like a magnifying glass. So I don't know if you've done this or whatever, but if you take a magnifying glass out in the sun, you can get it to make a pinpoint of sunlight on like a leaf or an ant, and it'll burst into flames. And this works the same way. So you walk out in the sun and you don't burst into flames. But if someone had a giant magnifying glass on you, you would. And so these chlorophyll Bs, their job is to focus the light energy on chlorophyll A. And I'm going to kind of show you how they do this in a more my screen share go. Where are you? So there you are. Okay, there we go. All right, well, I'm sharing my whole browser, but it doesn't really matter. So this kid, glued a bunch of magnets on here, or sorry, not magnets, why do I keep saying that? Mirrors, and you can see like he can use it to burn wood, right? Which wouldn't normally happen when you went out in the sunlight. And it's at that focal point, right? That one point where all the light energy is concentrated, and that's what chlorophyll A's and those accessory pigments, the carotenoids and xanthophylls and stuff like that, that's their job. And you can see that's a lot of energy, and the purpose of that energy isn't to set stuff on fire, it's to make those electrons jump. So you can see it can burn metal, it can even burn concrete, cut a can in half. That's a little daredevilish there. You'll lose a finger. But anyway, that's how photosynthesis works. So I'm going to go back to our PowerPoint. All right. And so chlorophyll A is the one that is getting all the light energy focused on it. And like I said, it excites those electrons. And we learned that in chapter 2. Chlorophyll B and the carotenoids, there are other things like xanthophylls and stuff like that, but they're grouped into this group. So there's really, 3. This is a broad group, but for this purpose of this class, we're just going to do 3. So chlorophyll A is a darker green color. It's actually literally the same color that the artist used to draw this. And chlorophyll B is a lighter, like more of a yellow-green color, and it's literally the same color that the artist used to draw this. And carotenoids and xanthophylls, accessory pigments, they're yellow. And when you looked at that YouTube video, you should have seen all 3 of those pigments, the more blue-green, the yellow-green, and then the yellow being separated out through that chromatography experiment. And so accessory pigments increase the wavelength that plants can absorb in. But one thing that you can see here is that this is the absorbance. And so this graph shows you that plants are very poor at absorbing light in the green and yellow spectrum here, and which basically tells you plants are greenish yellow, which you already knew that. Okay, so here's the chemistry behind it. I don't want you guys to get too caught up in this, but usually scientists, when we want to separate out things, we just throw it in a centrifuge because it's real easy to do that. But centrifuges aren't good at separating things that are close in mass, because centrifuges separate things based on their differences in their mass. And it makes sense. Heavier things go to the bottom, lighter things stay at the top. If we look at chlorophyll B on this particular structure, so I'm showing you this to let you know that chlorophyll is pretty complicated. That's what this is, and it has that fatty acid tail, which we talked about. And in chlorophyll A, this is a methyl group, so a CH3. Do you guys remember what CHO is from chapter 4? It's a carbonyl group. And so the difference in atoms here is essentially water. This has 2 hydrogens, this one is an oxygen. That molecular weight is 18 Daltons. Remember, a Dalton is like 10 to the negative 23 grams. So there's no way in hell that you could centrifuge out chlorophyll B from chlorophyll A. Just to give you an idea of this, so you hear on the news all the time that Iran is enriching uranium. So there's uranium-235, which is the radioactive uranium that you want to make nuclear weapons or to make reactor uranium. And then there's uranium-238. And so this is all mixed together in the soil. You'd go and find a uranium plant and you dig it up. But you wouldn't be able to tell the difference between uranium-235 and 238, just like you can't tell the difference between carbon-12 and carbon-13 and carbon-14. But the mass difference is 3, right? This is a little bit more. The mass difference is 18. This is smaller, but the way that they purify this uranium from this uranium is they use these, what we call ultra centrifuges. And these centrifuges are the whole reason that the U.S. is freaking out over Iran building nuclear weapons. These take years, so you have to spin this stuff for years at high speeds, hundreds of thousands of RPMs for years and years and years to separate U-235 from 238. So you could separate chlorophyll B from A doing this process, but it would take you, roughly 10 years and centrifuge that cost, well over $1 million. We don't really have time to do that in biology class, so we use that other way to separate things out, which is chromatography, which separates it based on its solubility and not its mass. All right, so the reason I'm showing you this is so that you can see that chlorophyll B and chlorophyll A are very similar to each other. They're only a couple atoms different. I'm also showing you this. I don't want you to memorize this, but I do want you to know that this is a magnesium center here. But you don't need to memorize this molecule. I also want you to see that this very much resembles a phospholipid, and it sits in the phospholipid bilayer of the thylakoid, which are these, remember these little green discs. So it's in this membrane. It's like right here in this membrane of the thylakoid where chlorophyll sits. All right, any questions about that? Okay, so we talked about this in chapter 2. And this works for any atom, but in chlorophyll, we have that magnesium that's in the center. It has 2 electrons. When light energy hits the chlorophyll molecule, and remember this is chlorophyll A, the electrons get excited and they go to a higher energy level, and the more energy we put in them, the better, the further away they go from the nucleus, and we know that electrons that are moving are called electricity. So we're generating electricity. Now, if we don't put enough energy in, they'll fall back down and they'll give off more light and more heat. But that's not the goal. The goal is we want to put so much energy in it that it flies off and goes down an electron transport chain, which is very similar to, say, a cord that's on your vacuum cleaner. So you can control the flow of the electrons. That should just be a review. Okay, so we have photosystems. These are light harvesting systems. They involve chlorophyll A. Remember, that's the main one that gets the light energy. B is like and carotenoids. These are like the satellite dish that focuses the light energy onto A. And they're assembled together in a thing we call the antenna complex, because it's like an antenna. So all these light green ones, the artist drew it as light green because that's the color of chlorophyll B. So chlorophyll B are all these light colored discs. And the dark colored disc, if you remember from that other slide, this is chlorophyll A. So the energy is being transferred onto chlorophyll A by all these different pigments, focusing all the light energy like the death ray, and causing the magnesium electrons to jump and go on to electron acceptors, which cause electricity. They produce electricity. So if we're going to go back to a satellite dish, we would say the dish itself is made of chlorophyll B and the carotenoids. The little receiver thing is chlorophyll A. This is going to get all the light energy focused on it, and then the electrons are going to jump and go. It's the same thing as plugging your vacuum cleaner into an electrical outlet. It flows from your outlet to your vacuum cleaner. And that's what we want to do. So a couple terms. This whole thing is called an antenna complex because it resembles an antenna. And the middle of this part right here, like on your satellite dish, you know, it looks like this. This thing is called the reaction center, where chlorophyll A is. So those are the terms that you need to know for this. All right. So when we look at the photosystems, we can break them down into 2 different kinds of photosystems. So this is that same slide that we looked at before, and we're focusing right here on the light reaction. So we're not looking at the dark reaction yet, just the light reaction. The light reaction consists of 2 photosystems, photosystem 1 and photosystem 2. Photosystem 2 was actually discovered after photosystem 1, and then they realized that this one came first in the chain and this one came second. So rather than renaming them, which they should have done, they just left it. So photosystem 2 comes before photosystem 1. If it was up to me, this would be called photosystem 1 and this would be called photosystem 2, but it's not. And so here's what's going on. Here's our antenna complex, this thing right here. Here's our reaction center. So that's a chlorophyll A, remember? These are all B's. B's focus the light on A's. When we get light energy on that, it excites the electrons and the magnesium. Remember, there's 2 of them to a higher energy level, and the more energy we put in it, the further away they go. And then they go off to a electron acceptor, which passes on to another one, and so on. And this is exactly like the electron transport chain in respiration. Different players, and remember in respiration, I made you know these. You don't need to know the names of these, right? We're just going to call it an electron transport chain. So light energy hits photosystem 2, excites the chlorophyll in this A. This particular one is called P680. Any guess why it's called P680? This one's called P700, by the way.
That's 680 phosphates.
No, but remember that we're talking about electromagnetic wavelengths. And so this reason this is called P680, P stands for photosystem. The 680 is where it's best at absorbing that wavelength of light. So P680 absorbs light best at 680 nanometers. So can you guess what P700 is? P700 is best at absorbing light at 700 nanometers. All right. So in here, the electrons get excited. They go through an electron transport chain. Meanwhile, this is photosystem 2. It has light shining on it. It's getting excited. Its electrons are jumping. So these electrons that leave are replaced by the electrons coming in. Does that make sense? Okay, I'm gonna try to summarize this real quick and then I'll end the lecture. But so these electrons are leaving. 2 electrons jump and leave, right? That means that this chlorophyll A has 2 less electrons. The whole system would shut down. We wouldn't be able to flow electrons if we don't replace those electrons. So the 2 electrons it lost are replaced by splitting water, and we put them back. Splitting water also produces protons, right? Remember, because water, if we take a hydrogen, it's actually 1 electron and 1 proton. So if we take 2 electrons from 2 hydrogens, then we have 2 protons left over, right? And that's this. And then what's the other byproduct? Is oxygen. So this is where plants give off oxygen. Now, those electrons have been replaced and light energy will excite them again to cause this chain to continue. So those 2 electrons are coming down this electron transport chain. If the electrons in P700 was still there, it would be like you trying to park into some parking spot that already has a car in it. So those electrons have to leave first. So they get excited, they leave. These electrons coming in from photosystem 2 replace the electrons lost in photosystem 1. These electrons then go on to the electron carrier, NADP plus, to make NADPH. Remember, this has 2 electrons, just like the 2 electrons on NADH and FADH2 in chapter 9. All right. So that is how the photosystems work, and we'll get into a little bit more about them next time. So don't forget, you guys, your test is due Monday by midnight. I'm going to jump over to office hours right now, so if you have any questions, this will be the last chance that you can ask me. And then I'm just going to do a review on my own as a separate Zoom call with no one at it, and then I'll post that on the website this afternoon. Any questions?
Nope.
All right, good luck on your test. I'll see you guys either in a couple minutes or I have office hours on Monday as well. You can hit me up there. Otherwise, we'll pick this up on Tuesday. So
Thank you.
have a good weekend.
You too.
Bye.